Hello and welcome. We're discussing where art means art, creative exploration of fertility research and treatment. And the hashtag for this event is Fertility Arts. PET is producing this event in partnership with New Horizons in Fertility Research, which is a project based at the University of Edinburgh's Wellcome Centre for Cell Biology and also at the University of Warwick. And it is supported by a Scott Penn Wellcome Engagement Award and Wellcome. And I'm Sarah Norcross, Director of PET, and I'll be chairing the discussion. How might creation of an engagement with artworks help patients, scientists, and artists to communicate with one another and to develop new insights into fertility? The project New Horizons in Fertility Research has been exploring this question in relation to egg cells and early human embryos. Bespoke artworks created as part of the project are being used to help patients in Edinburgh and Coventry to help them understand, visualise different aspects of fertility and infertility and related research. Meanwhile, scientists and researchers are seeking to improve their understanding of patients' views and experiences and to find new perspectives on research questions by developing and discussing artworks as part of this project. At this event, we have various people who've been involved in the project, including junior and senior researchers, a glass artist, a laboratory manager, and a poet. They'll all be presenting their work and discussing what they've learned and considering future possibilities. So let's find out more about the project and hear what they've been doing, and more importantly, to see some of these artworks. So our first speaker this evening is Professor Adèle Marston. Adèle is Professor of Cell Biology at the University of Edinburgh, and she's the Director of the Wellcome Centre for Cell Biology there.
Hi, as Sarah said, I'm Professor Adèle Marston at the University of Edinburgh, and I'm going to start by telling you a little bit about the science that we're doing that is associated with the art project that you're going to hear about from the other participants. So our science is part of so the art project is part of a scientific project that is also a collaboration between Edinburgh and Warwick, and the scientists you're going to hear from today are all working together on this project. And so the aim of our project is to understand why IVF and fertility treatment often goes wrong, and also why people need IVF in the first place. So what is it that is the cause of the faulty eggs and embryos that lead to these problems with fertility? And we're really looking at the very early stages in this process, and that surrounds having the right number of chromosomes, so the right number of the genetic material in those eggs and embryos. And so what we know is that in humans, there are very frequently errors. So up to 1 to 2% of all human sperm have the wrong number of chromosomes in them. And this is a high chance of having an infertility problem. And even in eggs, the problem is even much worse. There are up to about 30% of all eggs that have the wrong number of chromosomes. And this is a problem that also gets worse as women age. So you can see on this graph, when women are in their 40s, they actually have a very high chance of a trisomic pregnancy, which means a pregnancy with the wrong number of chromosomes. So you can see this more clearly here. So normally, humans should have 2 copies of all of the chromosomes, but what you can see in this example here is that there are 3 copies of this one chromosome, chromosome 21, and that's chromosomes from a Down syndrome individual. And this usually arises because either the egg or the sperm has an extra copy of chromosome 21. And so in our work, we're trying to understand where these kinds of errors come from. And just to give you a little insight into what we see when we look at these eggs and embryos down the microscope, which is one of the major ways that we're trying to understand what the problems are. We see eggs at different stages, at these very early stages, and then they go through 2 divisions. And what I want you to notice here is that they put some of the chromosomes into this very small structure called the polar body. And the egg will sit in this stage waiting for a sperm to come, and then that will generate the embryo. And the reason I want you to notice this, this polar body and this egg, which is at this stage metaphase 2 of meiosis 2, is because that's the stage that you're going to see in my glass artwork, which I will show you at the end of my presentation. And so we're very interested in understanding how the eggs develop through this process, and also what the chromosomes look like. So you can see the eggs on the top of this slide, but in the bottom we can see a zoom in on actually what the chromosomes are doing, and you can see that that there are many complicated things even on this slide that are going on. And these are the molecules that hold the chromosomes together, the spindles that will pull the chromosome apart, and really we want to know how all of these things work together to ensure we generate a healthy egg at the end. So I'm just going to give you 2 highlights of things that we found. So this is one of the first studies that we did in the lab, and again, this is a collaboration between our lab in Edinburgh and the labs in Warwick. And what we found is that a particular protein called here Shigoshin 2 or SGO2, which you can see on these chromosomes in the green here, in young women stretches across these what are called the kinetochores, and is quite robust on the chromosomes. But in old women, what you can immediately see in this image here, when a woman is in the 39-year-old woman, you can see that in this egg we lose this structure on the chromosomes. And we've looked at lots of these eggs, 52 of these eggs, from very kind donations from women of different ages, and you can see that as women age we have a decline in the number of the chromosomes that have this nice green bridge of Shigoshin. And we think that this is part of the mechanism that is responsible for the increase in errors with age. And then in another study, we're really interested in looking a bit later. So what happens after fertilization in those first cell divisions of the embryo? And as I've said, the sperm will come in, you have the zygote, and then the zygote will need to divide, which will eventually produce an entire embryo. We're looking at the very early stages, the very first divisions that lead to about the 8-cell embryo, and we know that there are also many divisions in this. And this was work actually led by Cerys, who you're going to hear from later, who has done lots of live cell imaging, so we can really look at this division in real time. And what you can see is that very often this division doesn't happen very well, and she has documented a huge number of errors. So this was quite surprising, but also very interesting. And we're now trying to understand what this actually means for later embryo development. And so I just want to thank all the people that have worked on this, particularly the Oocyte group here in Edinburgh and all our fantastic collaborators in Warwick. This has really been a great experience working with all these fantastic people. And then I would like to show you my art, which, as I will remind you, I was very interested in trying to depict what we can see down the microscope, and also add some sperm to this because we don't actually often see those. But here is a polar body with some chromosomes in it, and we have a nice spindle here with some more chromosomes on it as well.
So was it a fun process for you to do this? Oh.
It was very fun, actually. Yes, it was very enjoyable. I have to admit, though, that I did get quite a lot of help, because while I was making this, I had actually a broken collarbone. So I was having quite a lot of help from Sarah Jane and another public engagement professional, Muriel, while I was doing it. So I had the idea, but I had to get help with placement.
So the next speaker is Sarah Jane Judge, who is a public engagement manager at the Wellcome Centre for Cell Biology, based at the University of Edinburgh. So Sarah Jane doesn't have slides, so just which makes life simple.
Yeah, so I'm the public engagement manager at the Wellcome Centre, although I actually did my PhD at Warwick. So it's particularly nice for me to be working with my old university as well. Um, I want to tell you a little bit about the process of the actual project making the art. So obviously Adèle has covered the science behind the project, but how did we end up working with the artists and making these pieces? Well, as Sarah mentioned, this is a project that's been funded by the Scott Penn Wellcome Engagement Awards. So we were encouraged to look for opportunities to work with patients and involve them with our research. The issue we have as a centre is that, as Adèle has explained so nicely, we're doing very fundamental research. And so there's actually across our full Wellcome Centre, there's very limited opportunities to work directly with patients, where, for instance, some of the other centres, if they're, say, working with integrated parasitology, they'll be working directly with patients who are suffering from a parasite. That's not something that we have. So this was one of the few opportunities where I could see that we could directly work with people who were experiencing the issues that our researchers were looking at. Our centre also has a long history of doing glass art projects, because the previous public engagement manager to me was an artist. And in fact, our centre had a kiln and had an artist in residence glass maker for a while. And so many of our researchers over the years have had the opportunity to make glass that represents their art in some way. And there was a real keenness to keep that going, even beyond us having the position of artist in residence. And so we came up with the idea of doing the glass art project and asking for funding from the Scott Penn Wellcome Award, based on just a desire to get more hands-on and make more glass pieces. We applied for this. It feels like a lifetime ago. It was literally March 2020. I think you probably think of something else that happened that particular month as well. And in some ways it was great timing, and in some ways it was absolutely terrible timing, because for a project that involved people getting hands-on and meeting up and going and making art, that really delayed things. So that's how it's taken us quite so long to get to the point of now where we're able to share these glass pieces. We worked with David Mola because he was a local glass artist, and he was quite easy to find because his, as I'm sure he'd point out, his studio was called the Glass Lab, so it felt like a natural fit for scientists to work with. And from there, we worked closely with David. We worked with patients, fertility patients. There was a lot of focus groups. There was a lot of meeting of minds and discussions, and then that really helped to shape the artwork, the central artwork, which David will be introducing. We're very lucky to have our artist here tonight. And from there, it took a long time because of social distancing, but eventually the researchers from Warwick were able to come up, and we were all able to get together in David's lab and make these beautiful pieces that we're going to display for you. And the purpose of these really is to act as a talking point. It's to get people talking, to get have that object that can kind of almost be treated as a handling object to inspire conversations. Not all researchers are great at, initiating conversations with the public, but I've witnessed them being able to do that just through, people will come up and ask about the pieces, and then that starts a conversation. So we've held a couple of workshops already, various locations, including the big fertility show in London a couple of weekends ago, and we plan to do much more with this. We plan to have it on permanent display, but also to be able to take it out and meet people, meet patients particularly, and use this as a point of starting a dialogue about the type of research we're doing and how patients can have input into this and have the chance to ask their questions about what kind of research is happening in the fertility space.
And so now, following on from her, we have David Mola, the glass artist. And so he works on public and private commissions of architectural glass, and he is the manager of the David Mola Studio and co-director of the Glass Lab in Edinburgh.
Good afternoon. Thanks for having me. It's a pleasure to take part in this event. My name is David Mola, and I'm going to, my presentation is going to be very different probably from all of the other ones. I'm not a researcher, I'm a glass maker, and I'm here because looks like a long time ago, in 2020 actually, that I was just contacted by Maria, which at that time was the public engagement coordinator for Edinburgh University, to make a couple of pieces for this theme of fertility and research and treatment, and a series of workshops with the researchers. So I'm going to explain you a bit how was that process. It was a long and enjoyable but difficult process, because it was, as Sarah Jane mentioned before, just before the pandemic, so many things happened in between. We need to change the route on the way. And then I'm going to explain you just briefly how was that trip. The project, I call it the Inner Forest. And it comes in different phases. It came in different phases for how the timing came, how the circumstances were happening, but as well there are just different levels and different ideas. First of all, we started with a series of discussion with the public event engagement coordinator. We made a sort of brainstorming, and we came to many different ideas, but very soon it was one idea that it was growing bigger. And it was this place, this Svalbard Global Seed Vault. I don't know if you are familiar with that place. For those that you are not, it's a seed bank set in northern Norway. And the idea is that they stock just seeds from all over the world, just to being able just to recover it in case of need, in case of any need of just lost any crop or lost any variety of seed. And we found just a close parallelism with this and the fertility process. So we start just pulling through that first thread just to create an image. So then I started we start producing a series of twigs and buds, so all made with glass. So I'm going to explain you a bit briefly just the technical process on how we made the artwork. So we started just modeling the buds with wax or paraffins. From there, I take a mold made out of plaster and quartz, and then that's filled with glass and heat on the kilns. So then later on, when that has melted, I remove all of the plaster and quartz, and then I have the glass objects. Parallel to that, I was just collecting plenty of branches and did the same, just make reproduction in glass that then they will join together. So the idea is just to have some branches that it leads into a bud, into a potential flower or leaf, and some other that they don't. So yes, we made a collection of several just different ones, just to be able just to choose between. So that was just one part of the process. Another part of the process, it was very interesting, and it was a bit led by the pandemic, so we couldn't just join together with all of the researchers, which was the first idea, just to make one-to-one, just to get to know their places. So we needed to change it, and we had the time just to have a number of interviews with different patients. So for me, it was very interesting because I didn't have any idea of anything related with fertility. And then we just went through, I think it was in total between 10 and 12 different interviews with a variety of people. We were just interested in just have the most variety possible. We were interviewing men, women, people who was successful, people who was still in process. And from there, we were developing the second part of the piece, which was this base full of these sort of eggshells made out of glass. We came just with this sort of color, and it was reflecting a bit this sense of fragility, of complexity, also the idea of an egg. It was just very appropriate because of there was a lot of research on those lines. And in this case, we use a slightly different technique. These pieces are made with pâté de verre. So in that case, I use the glass as a powder. I mix it with a binder just to make a paste, and then I fill the molds with it. I heat it into a different temperature, and I have these sort of balls, sugar-like. On a third level, I wanted to show how complex all that is. No, just I think one of the things that I learned the most on this process is how many people are engaged on this process, from from the researchers to the patients to the nurses to all many different people, even just and now artists involved in there just to make understood more the process. And it was important for me to make it interactive. That was a bit of a challenge, because to make an interactive piece in a time where you cannot touch, you cannot meet people, it was a bit difficult. But I found that solution. The piece, on the plinth of the piece, there is two different sensor movements, and depends how the people moves around the piece itself is illuminated in one or another way. So just to make it just fully functioning, you need just two people moving on both sides of the sculpture, and then the piece start just, the illumination start just tilting together. So this last image is an image of the piece with different illuminations. One of the sensor is activate the light just hitting the box, and the other one is just light the base, the eggshell bottom part. When there is movement on both sides, both illuminations start just pulsing together. That is the main piece. Well, in reality, there was just two twin pieces. They are not the same. They are like twins with the same idea. I make two replicas. One, it went to live to Edinburgh. The other one, it went to live just to Warwick, to Coventry. And then after that is where the restrictions for COVID get a bit more relaxed, and we started doing all of these workshops with the researchers. So this is a bit of a brief picture of this process on these two main sculptures.
So our next speaker is Lucy Munro, and Lucy is a PhD student at the Wellcome Centre for Cell Biology in Edinburgh.
I'm Lucy Munro, a second-year PhD student in Adèle Marston's lab at the University of Edinburgh. So my artwork, titled The Bivalent, is a representative of one of the experiments that we use with the eggs that are kindly donated to us. So in this experiment, we use a process called chromosome spreads, and it involves popping the egg and letting the chromosomes out to then settle on a slide. And we can then stain and image these with fluorescent markers to look at different proteins, which may be hard to see when the egg is still intact because there's so much in the way. So creating these chromosome spreads, which is seen on the right, and this is kind of a typical representative image of what I'd see down the microscope. And they were a key part of my master's project that I also did in Adèle's lab. And when we were doing this art project, I thought the shapes and colours were really rather beautiful and translated well to art. So they were an obvious and kind of easy inspiration to choose for my artwork. So actually working with the glass, I think, really brought my data to life in an accessible way. So the kind of I think it was copper that we used for this blue kind of texture, and the way that the texture that it created, I think really represents kind of the chromosome spreads and actually the DNA itself. And then the green and red beads on top are kind of tactile, and they're a way to represent my proteins of interest. And then it's a way to then kind of talk about what these beads represent with the people who can touch them. And then how I think my piece kind of fits in with everything as part of the collection, I feel it kind of opens a discussion about talking more about how some of the experimental techniques that we use, and kind of like allowing the public to see into like just kind of like how pretty science can be sometimes, and it's not all those like white lab coats and sterile, like it's actually quite colorful and stuff that we do. And then kind of overall, the project has been a lot of fun to work on, and it's been really great seeing how different groups of people have responded to our work. So we've taken it out to a few places, and it's nice to have that interaction with them. And I think it raises awareness to others how important our work is to reproductive science, but it's also been a way for us to connect with the patients and hear kind of the other side and the people who are actually donating to us, and kind of talk together about our like kind of symbiotic relationship that created this project and allows us to do what we do. So that's kind of the inspiration for my one.
Okay, the next speaker from the Edinburgh lab even is Dr. Gerard Pieper. Gerard has been working in Edinburgh for a little while, actually. I think our paths have crossed before. And he is the Sir Henry Wellcome Postdoctoral Fellow at the Wellcome Centre for Cell Biology at the University of Edinburgh.
With my artwork, I've tried to highlight a bit of a different aspect of our research. So the work represents a few different stages in the life cycle of a frog. So we have the eggs on the left, which are laid in the water. Then in the middle, we have the moment of fertilization with a colorful, multicolored egg at the bottom and a sperm cell on top. And then on the right, we have the development into the adult frog. So I've chosen this topic because in my research, in addition to working with human eggs and embryos, I also study frog eggs. So I've used these three scenes that are centered on the fertilization event to show that even though many aspects of the life cycles of frogs and human are very different, many aspects of our biology are also very similar. So this is one of the reasons why it makes sense to study frog eggs. I mean, you look into the egg itself of frogs and humans and look, for example, at the way that the DNA behaves inside the cell, you can see that many of the basic cellular processes are actually quite similar. Another benefit of frog eggs is that they have a very unique feature. You can take out all of the components out of the egg and just put them in a test tube, and then the cellular components continue to function outside of the cell, which is very useful. And then you can use these cellular components as building blocks to reconstruct part of the cell that you are interested in. So for example, in my research, I tried to construct the small molecular machines that are attached to the DNA that are important to make sure that each egg cell has the correct number of chromosomes. So what I hope to achieve with this artwork is to make people aware of how by studying different animals, we actually get to understand more about the biological processes that underlie the way our own bodies work. And this is of course not just limited to frogs. Researchers have studied many different types of animals such as starfish and mice. And actually a lot of the knowledge that we have about egg cells at the moment has actually come from studying different animals. So I'm very glad to be able to translate the biological work that I'm doing on frog eggs to the few precious human eggs that are donated to us, so we can get the most out of each egg that we get. And through this project, I've really come to appreciate the human value of the samples that we work with, the human samples. So each egg cell or embryo is really part of someone's journey and story, and I'm very grateful for everyone who has donated material for our research.
Now we're going to welcome back Dr. Sarah Jane Judge, who is going to show us some of the tiles created by the researchers who haven't been able to join us this evening. You're not going to show them all, but just a little small selections.
There's been a lot of researchers involved in this particular project, so I wanted to pick a couple or a few extra art pieces that of speakers that weren't able to be here tonight, either because they have moved on into new research or just in terms of event timing. And I tried to pick some pieces that I felt were maybe a little bit different to the rest. So also had taken a different artistic slant, or were explaining a slightly different aspect of the research than perhaps some of the ones that you will see today. I'm going to start with somebody who you will see today, but who is being pre-recorded. And so you'll see them towards the end of our session. And this is the work here of Geraldine. And it's titled Breaking Free. And it is a human embryo at the blastocyte stage, so around day 5 of development. And it's hatching from its shell known as the zona pellucida. I'm sure the researchers will correct me if I've pronounced that incorrectly. And this here, the hatching cell in blue, has made contact with the underlying cells of the endometrium, or the womb lining, at implantation. The embryo is attaching at a gap in the endometrial cells, and the orange signifies molecular signals passing between the endometrium and the embryo as they communicate in preparation for implantation. Implantation is where assisted conception treatment most commonly fails, even when an apparently healthy blastocyte is transferred. And Geraldine has gone on to say that our research aims to understand why some embryos have a higher chance of normal development than others, which may arise due to chromosomal variations being generated as embryonic cells divide. And I think you can agree that it's very colourful and very eye-catching, and that mosaic effect has really come through. Another piece, again, I think this is arguably one of the most different ones. This is by Nigel Burrows. It's titled Crafting the Metaphase Plate, and it's entitled Chromosomes Captured by Microtubules. Nigel writes, this represents paired chromosomes, chromosome arms in cream, and the mechanical spindle with its 2 poles, which we see in the orange, that send out fibres, we see them as the red, to capture the chromosomes and reposition them in the cell. Nigel's work focuses on the mechanics of chromosome movement and changes in their internal architecture, that's the green and black dots, under the forces from the spindle. Nigel says he also works on human reproduction, hence the suggestion of insects and flowers there. And I think this one always reminds me a little bit of maybe Star Wars. I think the TIE fighters in Star Wars and the lasers, yes. So you can see the artists, although had a very similar brief and the same kind of materials available, David was able to support them to do very different things with their artworks. Finally, I want to show you one of the favourites. So I say it's one of the favourites in that this one is always the one that people will come up and comment on how beautiful it is and ask if they can buy it. And this was by Dr. Emma Ford. It's titled To Err in Mitosis is Human. And this shows a zygote during first mitotic division. Emma writes, we have recently discovered that the first division of human embryos is highly error prone. During the first mitosis, chromosomes are much more likely to be segregated into the wrong cells than in later divisions. Here we see the chromosomes, that's the colourful rods, are aligned in metaphase plate. However, one of the chromosomes, the top right in the red, is unaligned, meaning that it's not connected correctly to the spindle fibres and will likely end up missegregated. So you can see, very pretty. I understand why people would be keen to own that particular one. But yeah, it was lovely to see them all together. And in fact, originally the plan for these pieces that the researchers made was to just have everybody make theirs, and then they would take them back to their relevant institution. And when we saw them all together, and I believe you're going to get a chance to kind of see a slideshow of them at the end, we realised they couldn't be separated. It would be too much of a shame. And so we actually ended up commissioning David to make replicas. So the ones you're seeing tonight are all the ones made by the researchers themselves, but there is a copy of the Edinburgh ones at Warwick and the Warwick ones at Edinburgh, and we're keeping them as a full collection because they were just too beautiful to separate.
So now we're going to move on to some members of the team at Warwick who are going to share their art. So first of all, we have Dr. Muriel Erent, who is the laboratory manager at Warwick Medical School.
Hello, everybody. My name then is Muriel Erent, and I'm the lab manager in the laboratory of Professor Andrew McCange at the Warwick Medical School. My tile represent chromosome segregation, and as Adèle has already talked about it, it's during cell division, then going from one cell to two cells, the genetic material or DNA contains the genes, and the DNA is organized as chromosomes, which are here the colorful stripy things, and this DNA or the chromosome, sorry, has to be evenly divided between the two cells. And as Adèle mentioned, in case of error in the number of chromosomes, this is associated with cancer or developmental syndrome or miscarriage. And these chromosomes are here represented by the stripy, colorful lines, but they are moving to the edge of the cells because they are connected via the kinetochore, which are the beads in red or blue, and they move along the microtubule fibers, which are the brown line. They are made, in fact, of copper wire originally. And in the lab, in the McCange lab, we are looking at chromosome segregation in human cells. And I hope you can see the homology between the similarity between here in green, the kinetochore, and is what I try to represent by the beads in red and blue. And the on the right, the pink line are the microtubule fiber forming the spindle, and the DNA is the chromosome is in blue there. Then since we have all this expertise in chromosome segregation, since few years we have then applied this expertise to the human eggs and embryo. And this is the project we do in collaboration with the Adèle's group. And you can see here we look at the same elements, which are the chromosome, the spindle, and the kinetochore here, which are in pink. As mentioned before, I think these tiles are very colorful and attract usually people. They've been shown then in scientific conferences and in also other public events, and we usually, it's a very good conversation starter, and it's been really nice to be able to talk to different members of the public. Either people are just curious, come and we explain what we're doing, why we're there, or sometimes we have also we realize that the people have already donated material for research and scientific research, and they're really happy to realize how useful these donations are. Then it's really very privileged to have this discussion. And yes, this is my chromosome segregation tiles.
Following on from Muriel, we have Aleksandra Byrska, who is a PhD student at Warwick Medical School. So she's going to show us her tile now and explain the science behind it.
So my tile is called a mosaic blastocyst. As you can see, it's quite colorful, and it's very similar to what was shown by Sarah Jane before, made by Geraldine, because accidentally we had quite a similar idea. But starting with a little bit of science behind my tile, as explained before by Adèle and Muriel, a fertilized embryo divides quite quickly in the first days of development. So it goes from 1 cell to about 100 cells in 5 days. And the most important part of this process is chromosome segregation. And just to recap to what was said before, it is very important to get that process right, because the chromosomes are little bits of DNA that contain all of the instructions on how to make a human. So we need an exact correct number. Humans have 46 chromosomes. And during this 5 days of development, an egg creates this about 100 cell hollow structure known as the blastocyst. However, as it divides, if this process goes wrong, the cells that are made will have slightly different chromosome sets. Imagine one of the cells loses one chromosome and the other one gains one, then they are slightly different. And if they continue to divide, they will create more of different cells. And if another event was to happen, it could create even another population of cells. So we have previously discovered that this first division is particularly error-prone. So it is actually quite likely that different cell populations will arise. And this is what I tried to capture with my glass tile, because it represents what we call a mosaic blastocyst. So a blastocyst that consists of different cells containing different sets of chromosomes. And I found this was an actually very cool idea, because I made a mosaic of a mosaic blastocyst, as a mosaic is originally a piece of art created with different pieces, colorful pieces of glass or stone. So this was my initial idea behind this. And going back to my tile itself, the blastocyst in my artwork is hatching, meaning it escapes the protective shell of zona pellucida. This allows it to grow, as zona is a bit restrictive in terms of the size, and also implant into the uterine womb. Before, it couldn't interact with the cells of the womb lining, but after the hatching, it will be able to. So in the blastocyst, we have 2 main cell populations: the inner cell mass, which is depicted with the colorful red, yellow, and blue little beads of glass. And this is what will give rise to the fetus and then the baby. And the blue surrounding cells are what we call a trophectoderm. So this is the population that will create the placenta and all of the extra tissues that need to be there to sustain the pregnancy. And what is particularly interesting, by this research we're doing, we have learned that about 80% of all human preimplantation embryos are in fact mosaic. So this seems to be a normal state for them. But we don't understand what this means for the development, because some of them, as it was mentioned before, aneuploidy we associate with adverse outcomes such as miscarriage, or it's even implicated in cancer. However, some of the embryos that are mosaic and contain aneuploid cells will develop normally. So this is what we're trying to understand in our lab, hoping that this will help to advance the IVF process, improve success rate, and support clinical decisions. So yeah, as said before, this was a really cool project. I had fun creating this, and I'm really grateful for this opportunity. It's a great conversation starter for the patients, as it illustrates how beautiful the science is, and gives people with maybe less science background the idea of what we do and how important donation of material to research is.
Next have Dr. Currie, who is a postdoctoral researcher at Warwick Medical School, and she also works with the Coventry Centre for Reproductive Medicine.
So yes, my name is Cerys. I'm a research fellow, so a postdoc at the University of Warwick, and I'm going to talk to you about my tile, which I labelled not particularly creatively, called microinjection, as this is the experimental technique that it's depicting, and I have my tile here as well. So I wanted to talk a little bit about what the structures of the egg that I've captured in my piece. So I would, at this point in my research, I was working on immature eggs. So this is a GV oocyte, so it hasn't yet undergone meiosis. So in my piece, I captured the zona pellucida here in pink glitter. And I thought that was particularly nice because it does look quite fuzzy. And actually, when I was looking at these eggs down the microscope, it did look like this. Here I have the germinal vesicle. So this is the nucleus where the DNA is. So it is circular. And I've captured the DNA as well here in these little rod shapes. But there are circles within here, as in a GV oocyte, the DNA does kind of arrange itself into a circle. So it's a really beautiful structure. Here in blue, I have the cytoplasm. So this is like the liquid inside of the egg. And here I have the microinjection needle, and I put this in copper wire into my egg. So this is what a actual germinal vesicle oocyte looks like under the microscope. So you can see I've overlaid the structures that I've shown in my piece onto this image here. So here's the germinal vesicle, the nucleus, it's a circle here. The DNA will be inside. The cytoplasm is here. It looks very grainy. And the zona pellucida, hopefully you can see that kind of like fuzzy type structure that I've tried to capture with the glitter. And then to the right, you can see the microinjection process. So I was, at the time that we made these artworks, I was doing this experiment a lot in the lab. So like Lucy, this was the inspiration for my piece, just because this was what I was seeing down the microscope every week. So you can see we have a holding needle to hold the oocyte in place. And then on the other side, we have the much thinner microinjection needle. And this is a picture that I've taken down the microscope while just before performing this experiment myself. And here we have a human GV oocyte that was donated to our research project. So in my piece, I've captured the needle with this like very big triangle. And then I overlaid it onto the oocyte. So hopefully this explains the science behind my piece. And I wanted to say, for my piece, I was hoping that my piece would open a discussion around the experiments that we're doing in the lab, because I think the needle itself looks quite abstract in my piece, and I was hoping that it would catch people's attention and people could say, you know, what's this random triangle doing here? And then I could explain the microinjection process and talk about the work that we are doing in the lab. And finally, I want to thank all the patients who donate their material to research. I think I speak for all of the researchers when I say every single egg and embryo that we receive for research is incredibly precious, and we treat those with the utmost respect. A lot of these experiments are very tricky and delicate, but we always are putting in our absolute best efforts to make the most of these precious samples.
Thank you very much, Cerys. And I think it was, I'm going to quickly ask you a sneaky question, actually. So it was your ambition by doing an abstract needle to make it a talking point. Did that tactic work? Did people ask you about it?
It has worked sometimes. Yeah, people do ask me. I think most of people's attentions are captured by some of the more like colourful, particularly Aleksandra's piece. A lot of people like this one, but a few people have asked, you know, what is this random thing sticking in here? So yeah.
Great. Okay. So the last member of the Warwick team to speak is Professor Geraldine Hartshorne, who is Scientific Director of the Coventry Centre for Reproductive Medicine. She's a Professorial Fellow at Warwick Medical School, and she originally completed her PhD under the supervision of the IVF pioneer, Professor Sir Robert Edwards.
Hello, everybody. I'm here today to encourage anyone who wishes to donate their genetic material to research. All progress since the very earliest days in IVF has really relied upon the availability of human genetic material for research. Animal research just is not the same. Researchers are working on different areas and they will need different things for their research. Now, our group is studying events just before and just after fertilization through to the second cleavage division, which is when the embryo has reached about the 4-cell stage. In my job, I spend a lot of time and energy looking at embryos that belong to people who haven't become pregnant during their IVF treatment. I'm attempting to understand what's happening with those embryos and how we can develop bespoke treatment programs for the people affected. And it's very clear to me that the problems mainly occur right at the start, just as the fertilized egg is trying to become a 2-cell embryo. Now, this stage is difficult to study. Firstly, because eggs are large, they're primed, ready to go through the fertilization process, and they're unstable. So they're at a sort of a difficult stage in their own history. It's difficult to access this sort of material. It's very precious, very special, very rare. And so ideally, what we're trying to access is human eggs at immature and mature stages and very early embryos to enable us to do this research. At the moment, if patients consent, what we're asking for are unfertilized eggs, eggs that didn't fertilize when we tried to fertilize them. So these would be about a day old. And because of that, they're not in prime condition. They might have deteriorated a little bit. So we use them, but they may not be top quality research material. Immature eggs, again, these are eggs that are collected that can't be used in the IVF process. Ideally, they would have been mature at the point when they were collected, so we're not sure that their response is actually going to be normal, but we can still use them and we can still learn from them. And we also use abnormally fertilized eggs, such as triploid or tripronucleate embryos. And these can also provide some useful information, but it's not normal fertilization. Our research has been criticised in the past because we were not using top quality material, so the results may not be reflective of the normal situation. To overcome this, we set up an egg sharing to research programme. Now, this allows patients who are young and with a good chance and probably having plenty of eggs to choose to share some of those eggs with us as researchers. And in return, we will pay for a large part of the treatment that those patients are going through. That payment is from our research grant, so it's all approved through ethical committees and so on. And this would allow us to access fresh, mature eggs, normal eggs from young women, which ought to be top quality material that we can then use in the research. We can fertilize them to make early embryos. And through doing this, we've gained some really exciting results about the first cleavage division that I'm sure you'll be hearing about. And this is helping us now to explore methods that might be able to help eggs through that first cleavage division that might allow us to improve IVF for the future. Now, the patient pregnancy rate from these egg share cycles is good, so we're pleased as well with that we're not disadvantaging anybody who is offering us eggs as part of this programme. And we've shown that even these fresh, mature, normal eggs from young women are still prone to errors, and the same sorts of errors that we were seeing in our previous research. So in that way, we've validated a lot of our previous work, which has also been extremely valuable. How can people get involved with this? What can people do to help? We really would value further collaboration and further people being involved in our research. And there are different ways that people could become involved. So first off, there are many people, young women often, going through the process of egg freezing, and their aim is to use those eggs in the future to become pregnant. Now, many young women will in fact find that they can become pregnant without the need to use those frozen eggs. So it's likely that in future, those eggs may no longer be needed by those patients. It would be great if clinics could provide information to those patients about the opportunity to donate those eggs to research if they no longer require them. People who'd like to do this, any patients out there who might be listening, you could encourage your clinics in this, and you could also contact us directly if you wish to. If clinics would like to collaborate with us, please consider whether you might be able to collect any immature eggs from patients who are going through the ICSI process and vitrify those so that they could be shipped to us at a later stage. This would obviously require patient consent, so we could help with that, and any lab consumables that may be involved, again, we can help with that through our research grants. Do let us know if you might be interested, and we can talk about it and find a way that would work for everybody. We're also keen to receive frozen embryos, but for us, because we're working right at the beginning of embryo development, we're really only most interested in those that were frozen at the 2 pronucleate stage. Most embryos nowadays are vitrified as blastocysts, but if anyone out there is still using or still has available those very early embryos that we used to freeze, please consider whether the patients might wish to donate those to research. And finally, we can accept patients onto our Egg Share for Research programme. Now, this is only taking place in Coventry. Any patient who wishes to consider this programme would need a GP referral to Dr. Reena Agarwal, who runs our egg sharing programme. She's based at the Coventry Centre for Reproductive Medicine. It's an NHS clinic. So for the future, the best way forward would be for there to be a tissue bank that manages all of the available material arising from IVF so that patients who wish to can donate their material very easily. Researchers who need to use certain types of material can access it in a carefully controlled manner. Banks are already available for all sorts of different types of material, such as stem cells or fetal tissue, cord blood, and so on. But there's none for human eggs and human embryos. And the reason for this is because at the moment, the law doesn't allow us to gain generic consent that would allow, say, some eggs or some embryos to be used in a range of different research projects. Each particular project has to be signed up to by each individual person involved, which involves a lot of work. It's quite a cumbersome system. It would be great to have a centralised bank so that people up and down the country could donate material if they want to use in research. It would streamline the whole process for clinics and for researchers. It would make a much more consistent patient experience of accessing and interacting with research. There'd be more research material available. There'd be fewer hurdles for researchers. Not that there shouldn't be some, but there should obviously not be more that are going to slow up the research and prevent the progress being made. And there should be careful, transparent regulation around the tissue bank. Donating material to research is always voluntary. There's absolutely no obligation. And for people who don't want to do it, that's absolutely fine. All the research that we do in this field is licensed by the HFEA, and it's carefully monitored by the organisations that we work for and recorded through annual reports, for example, to the research ethics committees. This particular project that we're working on now has been running since 2004. And that's 20 years now and counting, so we will be continuing into the future. And over those years, we've had literally thousands of patients who have provided their material to us. And I'd really like to thank each and every one of them for the time and effort and dedication that they've shown to allow us to do the work that we do to hopefully help and make progress. Clinical practice is working right at the limits of scientific knowledge in this field. There are many questions that we don't know the answers to. The only way to make progress, to improve the scientific knowledge about embryos, to improve the success rates of IVF, and to help individuals with their specific fertility problems is through research. And I hope you will support our efforts in this way. Thank you.
So the final speaker this evening is Robyn Kerr. Robyn is a writer and a hotel manager. She is the author of the novel Failing Adult, and tonight she's going to be telling us about her involvement in this project and sharing a poem with us.
Hi, so yes, I'm Robyn, and I did my placement at the Wellcome Centre for Cell Biology during my third year of university. And then while I was there, I worked with a few of the researchers. I went through the labs. We've seen all the pieces that was made, the glass pieces that we've seen tonight. And I got really inspired by everyone's stories, the process, the amount of work and effort that goes into it to help people conceive after everything they've been through. So then I did a lot of personal research into people's personal struggles, the women and the men. and the doctors and the nurses and everybody involved, really. I've got a few people in my family that have been through it as well. My cousin, she went through IVF and then had twins. One of the people actually on my course who read the poem that I'm about to read, I didn't realise that she'd been through IVF and was unsuccessful after about 4 tries. So that also helped me want to do more. So I will start now. So it's titled Mother in Waiting. Crying in the car, still no blue line. Empty. It hasn't happened. Again. I had let my hope take over. Lists of names on paper. Colour schemes in my head. Dreams of a baby reveal. Is it me? Something I've done wrong? But I do as asked. Take the pills by the dozen. Tests in the hundreds. Injections I withstand. Supplements I endure. I have done it all. Not only once, a first, then second, a third, then a fourth. Now on to the 5th. Virology, collecting, freezing. How many embryos have made it this time? Side effects becoming my norm. Bleeding, pain, bruising, bloating, cramping, torn down, fallen apart. How many times can I go through it? I wish I hold, love to give. When will the time come? When will someone call me mum?
In different ways, Sarah Jane, have you established where a permanent home for these artworks will be? You know, when they can, where they can go and see them, that sort of thing. So
obviously, as explained, there's actually 2 full sets now for each of the locations, so it will vary a bit depending on whether we're looking at the Edinburgh set or the Warwick set. I believe Warwick has more of a natural space to display them in a new building that's opening, so I think that's a work in progress to get them displayed there. Our central artwork has been on display at Edinburgh for a while now, so it's in the front window of the Michael Swan building at Edinburgh University, where the Wellcome Centre is based, and we are now looking at a way to display the tiles as well. But just to kind of reiterate, however we display them, they will always be able to be taken out for these important workshops with the public and with patients as well. So we will look to have something, let's say, semi-permanent for them, that they can be on display but then also be taken down and taken around in a safe way as well. But yeah, more to come on that. Hopefully, when we've a little bit further down, we know what kind of space we have available.
And I know you took them to the fertility show at Olympia at the beginning of May. And what was the reaction like there from the public? I think a few of you, perhaps on the call, I think did Aleksandra attend that as well?
Yes, I was there.
And it's a sort of tricky environment, isn't it, the fertility show, because it is like a big commercial exhibition but aimed at patients. So I would imagine your stand, I wasn't there this year, but I have been virtually every other year, was pretty unique in that you weren't trying to sell anything to anybody. You weren't trying to get their email address so you could follow up and hound them about something else. And you weren't giving them, free medical advice. So so how did it go?
We have been asked many, many times if we're selling these because people were really keen on buying some. So as much as we'd love to, we only have 2 copies, so not at the moment at least. It was a great conversation starter, and it did catch people's attention. This was a very different environment to what we usually work with, but I found it very positive and very open, and people were willing to share the personal stories, and we could use the glass pieces as like explanation of what we're doing and make a reference to their own IVF journey. So it was a really good time, and I really enjoyed it.
Sarah Jane, how did you find it?
Yeah, if I can share one particular story that really, that I think kind of encapsulates what we were aiming to do, and maybe even beyond that. So people would come over just to ask what we were doing, because as you say, they're used to trying every stall trying to sell them something, and they would come over and ask about the art, and that would then start these conversations. And it was just towards the end, actually, the second day, somebody came over and asked about the, you know, how they could potentially donate eggs. And we heard a lot of people saying that they didn't tick the box to donate genetic materials when they went through their consent forms, because they didn't understand what they would be used for, and they didn't understand, you know, the importance of it, and they were worried about them being just disposed of. And, you know, that this was a great opportunity to kind of rectify that. But one particular person asked, well, if I did want to donate, you know, I'm convinced by you guys and what you're saying, but if I wanted to donate, how much would I have to pay to have it shipped to you? And I guess the researcher looked a little confused, you know, why would you pay? Oh no, we would cover that. Oh, but then surely you've got to wait for there to be X amount of eggs available before it would be worthwhile you shipping them to Edinburgh or Warwick. And we were just able to like explain that they're so precious. You know, we would absolutely pay the fee for the cost to take just a single egg somewhere. And I think that really resonated. You could tell with the person we were talking to that that this egg is as precious to us as it potentially is to them as well. And that, you know, it really made a difference, and they assured us that they would absolutely be ticking the box next time to donate, because you know, this is something that is so valuable to research. It's so valuable to our researchers and to the forwarding of science.
That's really ties into another question. It sort of answers it really. Could the artworks be used to help encourage patients to donate or eggs or embryos to research, as discussed by Geraldine Hartshorne in her, you know, very powerful presentation about donation to research? And so it seems that you know, from what you've said, that is an opportunity, and perhaps that also it's sort of uncovered from that, you know, I know we shouldn't base all how we go about things on a case study of one, but it is very interesting, isn't it, that, you know, they didn't know what to do. They'd got all these questions, and I suppose, you know, when you're in the thick of fertility treatment, that is, you know, that's not your primary goal, secondary goal, or probably even your, you know, third or fourth goal, doing that. So whether the information is provided or not, you might not take it in, or if it's not provided fully, you've got so many other questions about your actual treatment that you're perhaps not querying them. So there could be an opportunity there. I'm trying to think about how something like that could be used to help to stimulate those discussions in clinics as well. So
I think it's about answering, it's just about creating opportunities for conversation, for dialogue. Like you say, we wouldn't have been at that particular event if we didn't have the exhibition to showcase. So just by having the exhibition made us go into that scenario where there was this availability to talk to people. And we'd love to do it more and in different formats. It doesn't need to be at the fertility show, but having it will hopefully inspire us and cause us to go out and make these conversations happen.
Great. Okay, I've got a question for David now. Did you have the opportunity to visit the seed bank? And if you did, can you tell us what impression it made on you? So did you manage to get a trip to Norway?
I wish. No, I have never been. I will have, all that happened just when we couldn't move. So it was all our research locked in my house. So no, I have never been able to be there. As far as I know, I don't know much about the project, but what I know is that it's not open to the public. You can arrange a private viewing, but it's not that easy.
Can donations be made from clinics in Ireland? Would you accept their eggs and embryos? I don't know who that's a question for. Yeah, Aleksandra?
So that would be a question for Geraldine, but since he's not with us, I am not sure, but we will need to look into the process of donations in Ireland. But I think that should be possible to be arranged. But if you want to donate anything to research, please get in touch with Professor Geraldine Hartshorne. I think there are her contact details in the chat, and she will be able to help you. Thank you so much for considering donations to our projects.
Adèle, do you happen to know the answer, or are you happy to leave that to?
Yeah, I think Geraldine will definitely know more about that.
Great. Okay, so Aleksandra, we have a question for you now. So could it be argued that it would be good if the reproduction of these artworks were slightly imperfect? Then we could have mosaicism in copies, the mosaic representing mosaicism. I can't even say the question. It's I hope you can sort of get the idea that the question is trying to, that would that be another play on both the art and the creation of art, and also the biological process of mosaicism.
Yeah, that's a brilliant idea. I actually think that I've seen the reproduction and my own artwork next to each other, and they were slightly different just because, as Currie said before, we arranged the class, but they weren't ready until we left. So they will be slightly different. Also, I've managed to pull one of the beads of my artwork accidentally, and I'm not sure I placed it in the correct way. So that's also a difference. But in my mind, each color represents each karyotype. So each set of chromosomes for that particular cell. And since we have 48 chromosomes, the combinations are really endless. So we could have all different rainbow colored embryos, and that would perfectly visualize how unique humans are and how different all of the embryos are. So I love that idea.
Okay. We'll go to Gerard first for this one. For this is one for all the researchers. Has making these artworks made you think differently about what you're researching? Has it had, is it, I can imagine it was a great creative, fun experience. But then what did you take back to the lab from it? So Gerard, if you go first on that, and then we'll go to perhaps to Cerys and Lucy.
Yeah, I think actually making the artwork was a lot of fun, but I think I got the most out of it when we actually took the artwork to places, and we're talking like this London event, and we were actually talking to patients that came up to us. And I think this is where I got the most out of it, realizing that the material that we are getting, it's really a bit different than some of the animal materials that we get. For example, they're very precious to the people that donate them. There's a lot of feelings associated with them for them, and I think that was very special to see, basically. So I think that's where I got the most out of it, basically.
Yeah. And how about you, Cerys?
Yeah, I agree with Gerard. I think my perspective changed a lot, definitely when using these pieces to speak to patients about their experience of fertility treatment. I think that was really, really powerful and really important to hear those stories. I think it also helped, it changed my perspective to kind of see everyone's pieces together, because when you're just focusing in research, you're usually focusing on your experiment, your project, and you kind of get the blinkers on what you're doing. But it was really nice to see everyone else's pieces and the kind of like get that big picture view of all the real breadth of the project that we're doing and the number of different people that are involved in this project.
And how about you, Lucy?
I don't think I can really add much more than Gerard and Currie. I completely agree. Kind of like, I don't think our research changed so much as our perspectives on kind of like the material that we're using. And I think Cerys brought up an interesting point of like this artwork also kind of brought us together in a way so we can share our projects with each other, and then our research develops from that. So it's just a real way that's like kind of connected us, and then also connected us with the patient. So it's been really great. I think those two summed it up a lot better than me.
No, not at all. Someone's said traditionally images of cells and tissues have been 2D, but in recent years there have been advances in 3D images of cells and tissues. And has this development changed the way that researchers think about and visualize eggs and embryos? And if so, was it reflected in the artworks that you created as part of the project? So should we go to Adèle and then perhaps to Muriel for this one first, and then others can chip in?
So I think that's a very sort of great comment, because of course eggs are really big. So actually to image them, we do need methods that can visualize things in 3D, and we essentially have to do that by taking slices all the way through and then reconstructing that on the computer. So it is a very important thing for the research. And I think, you know, the chromosomes, when we're working on the chromosomes, they form such a small part of the whole egg that such methods are really, really important. Yes, it's completely critical for our research. In terms of the glass, I think, of course, you know, creating glass art in 3D would have been amazing, but, you know, we were there just for one day. And David did an amazing job in teaching us these techniques, but had we been blowing glass in 3D, I think that would have been, you know, a slightly longer learning curve. So yeah, but, you know, maybe something for the future. And obviously we do see that in the piece that David made as well.
Yeah. And Muriel, what did you think about that? You know, the sort of...
Yeah, I think I was thinking David is completely in 3D plus the light, and that's definitely different dimension. Our pieces got several layers still, not completely, but that could refer to, as Adèle was talking about, we need to do in microscopy, we do different images in 3D, then it's layers and layers of imaging. And we do study, I mean, the pictures I've presented and others, they are in fact just projection in 2D, but they are in the computer, they are 3D, and we can reconstruct and look through the samples, through the imaging. And all the tiles have different layers. We did different layers of different glass, and they have 3D structure. I mean, you have the beads, you have the different pieces of glass, plus the copper in between the two glass, which is trapped. And there is some aspect, not very much, but a bit of 3D going on, relief at least. It's not smooth.
And David?
I think, well, the pieces are not completely flat, as Muriel said. It's a bit of a bas relief, so it has a bit of relief. And I think it was very well used in many of the pieces, the actual quality of glass, of being transparent. So it's not 3D, doesn't has a volume, but in some of them, just you can see how you can see through. So it gives a very good impression of being with volume, with being just one thing behind or inside another. So it was a very good use of the material.
Great. So I think that's David's way of giving everyone a sort of an A star for their art there at the end, which is nice after Gerard's earlier comments about, oh, I hope I'm not going to be graded. Okay, so before we just go to Robyn, I think you sort of bringing some of the points together. I think it's clear that this has been a really successful project on so many levels in terms of, you know, inter-lab collaborations, in terms of bringing different people in to discuss and have awareness of the issues, and in terms of a legacy in these ongoing talking points. And also, if only you'd known they were going to be so popular, you could have been, you know, and there's a demand to buy them. You know, you could have made some money as well as have your grants. But quite, you know, quite exceptional, really. And I think, you know, to generate those sorts of conversations, that's something that is can be so emotionally charged as a fertility show as well. It is really fantastic. I mean, because at PET we come across and hear about patients who want to donate their embryos or eggs to research but can't, because the clinic that they're having their treatment as doesn't facilitate that. And so a research egg embryo bank that Geraldine mentioned is something that PET is very much in favour of and wants to campaign for once, you know, as the law is reformed in this area, to make it as well simpler for patients to consent to donating to research, because for those that do, we shouldn't be putting barriers in their way, because they often, they've been through enough with their fertility treatment journey. They don't want complex consent. They don't want to be asked about lots of different things. Often what they're actually seeking when they donate to research is closure, and not to sort of keep having sort of unhappy times in their lives reopened again and again, but to think this is the end of something. But now I think we'll go to Robyn to read her poem to close out the presentations.
2 seconds. Okay, so this second one is titled Hope. How long can one hope? How many breaks can one heart take? It didn't happen this time. It wasn't meant to be. One more round, one more try. Maybe this time it can happen. Maybe this time it will work. Hope is what I have. Hope is all I have.